Ladies and gentlemen, I am Wiktoria Mrozek. I am a researcher and biological data analyst at the Nencki Institute, where I obtained my PhD in neurobiology. I have an interdisciplinary background, including bioinformatics and biophysics, which come in handy in my work. As a fun fact, I can tell you that I am a part of the organizing committee of this Ultrasonic Vocalizations Conference since the beginning. And I am elated that we managed to grow this much over time. And each time we have bigger audience than before. So thank you all very much for being here. And let me now start my lecture. I hope you can see it now. Okay, did it switch?
Yes, it did.
Okay, so officially, hello again, everyone. So my talk today will be about a study that was meant to examine the role of serotonergic and glutamatergic co-transmission in the amygdala in the state that is a part of addiction process, specifically during the re-exposure to drug-associated context. So today, hopefully, I will be able to tell you a complete story of how when we begin with measuring the ultrasonic vocalizations, that can lead you to eventually a large portion of the biochemical network of the brain. The presentation will go as follows. So, as a quick intro, you probably all well know that the opioid addiction is a global public health problem. It is estimated that in 2021, approximately 60 million people worldwide were using opioids, and these substances accounted for the majority of drug-related deaths in 2019. Among all the countries, the United States faces the largest problem, and although the scale of the crisis in Poland, for example, differs, problematic opioid use still poses a significant challenge for our country as well. Regardless of the scale of the problem, a universal clinical challenge is a high risk of relapse, which is often triggered by environmental stimuli associated with prior substance use. Besides those two, we focused our research on the phenomenon of incubation of craving, which means the intensification of drug craving during the withdrawal periods, which additionally complicates and oftentimes prevents full recovery. And as illustrated in the addiction cycle diagram, even after a period of abstinence, when drug craving emerges, often reinforced by the incubation of craving, contextual stimuli such as places, situation, and people associated with the drug can trigger an irresistible urge to relapse. Therefore, understanding the neurobiological mechanisms underlying this response to a drug-associated context is crucial to develop more effective relapse prevention therapies. So here is the sketch of the study done by Adam Hamed and Miriam Kursa, the study which was followed by my research. So long story very short, they administered morphine to rats in a specific cage and then tested the context response to that cage without the morphine administration. The important takeaway for my study from here was that after the morphine sessions, the withdrawal period, can you see my pointer? Okay.
Yes, it's visible.
Okay, so the withdrawal period was necessary for the appetitive ultrasonic vocalizations to appear during testing. So if the tests were done the day after the last morphine session, there were no ultrasonic vocalization response. It appeared only when given the 14-day withdrawal period. Moreover, what they found from doing a big biochemical panel of analytes in chosen 7 regions of the brain was that first, the amygdalar levels of serotonin correlated with amygdalar levels of glutamate and vice versa. The levels of glutamate in the amygdala correlated with the serotonin in the amygdala. And that the levels of serotonin in the amygdala correlated with the number of ultrasonic vocalizations emitted during the tests. So at the moment when I was starting my PhD, we already knew from literature that amygdala plays an important role in emotion processing and stimulus-reward association. both serotonergic and glutamatergic systems have been previously linked to addiction processes in some form or another. And from the previous study done in my former lab, we knew about the correlations between the levels of those neurotransmitters in the amygdala and the other correlation of serotonin levels with the positive affect measured by the number of appetitive ultrasonic vocalizations emitted in response to the morphine context. However, the causal contribution of the co-transmission of these two systems in the amygdala to shaping the drug-associated contextual response remained unknown. Therefore, we hypothesized that serotonergic and glutamatergic co-transmission in the amygdala modulates the affective state measured by the appetitive ultrasonic vocalizations emitted in response to the morphine-associated context after withdrawal. And this served as a starting point of my work. So here, as you can see, the scissors, they symbolize the chemogenetic decomposition of the co-transmission of serotonergic and glutamatergic systems in the amygdala, which I were ought to do at that point. And we wanted to analyze the following thing. So first and foremost, the biggest aim of the study was to examine the effect of selective chemogenetic manipulations of serotonergic and or glutamatergic neurons. I hope that will be clearer soon. In the amygdala on contextual behavioral responses as measured by the number of ultrasonic vocalizations and the distance traveled. I will not have the time to discuss the locomotor activity results. So if you are interested, maybe you can ask me about them during the questions time. I apologize for that. Okay, secondly, we wanted to characterize the neurochemical substrates of the observed behavioral changes by analyzing the levels of 16 analytes in 7 selected brain structures. Then attempt to identify the neurochemical systems underlying both de-reduction and intensification of affective responses, if any, of course, and analyze the neuronal population activity at the end and the start of the contextual response test with catfish method. So the experimental design was as follows. And as said in the premise of my lecture, we began with the ultrasonic vocalizations. So after a period of initial acclimatization, we performed a tickling procedure aimed at assessing individual affective reactivity. Shinpei has already explained that to you today, but in case you were absent, then tickling is a procedure mimicking the natural form of play, where you, the experimenter, quite literally tickle the rats and you can measure the emitted ultrasonic vocalizations, if done correctly, mind you. And they should be in the 50 kilohertz class, so an indication of the positive affect. And then we perform the neurosurgical interventions. So by using dread viral constructs, we introduced the dread receptors into the amygdala. This hemogenetic technique in general allows for precise and reversible activity control of selected neuronal populations. And in our case, activation or inhibition of serotonergic and or glutamatergic neurons. So we were able to create all possible combinations of serotonergic and glutamatergic system activities in amygdala using the dreads. Thus, we created 8 experimental groups plus the 2 control groups, which are in the middle here, which were without the dreads. One received morphine and the other saline. And this experimental design enabled a systematic decomposition of the studied co-transmission. So if you remember the scissors, here is how we created them in practice. After the post-surgical recovery period, the animals were subjected to a 10-day place conditioning procedure during which they were given injections of morphine or saline in case of saline control group in a specific context, so a different distinguishable cage. Then we introduced the 13-day withdrawal period to induce incubation of craving And on the 14th day, 30 minutes after systematic administration of the DREAD ligand, which specifically activate just the DREAD receptors, otherwise neutral for the organism, we did a context response test. I mean, the animals, of course. And during the test, each animal was re-exposed to the environment associated with the morphine administration. Of course, we recorded the behavioral responses throughout the duration of the test. And then we did the quantitative measurements of 16 selected analytes via mass spectrometry. They are listed on the bottom right. And neuronal activity analysis in the 7 chosen key brain structures. So the amygdala, nucleus accumbens, striatum, ventral tegmental area, medial prefrontal cortex, hippocampus, and retroscranial cortex. And finally, we tried to link the data to the collected behavioral results. Just a second to zoom in. These were the viral vectors construct we used in the study to create these 8 groups. And of course, the most important part of my study were the ultrasonic vocalizations in the 50 kilohertz band. So short reminder for people maybe not that familiar with rats, 50 kilohertz ultrasonic vocalizations are widely considered to be an indicator of the positive affective states. And they appear in response to positive social interactions, natural or pharmacological rewards. And moreover, their emission is also caused by the anticipation of appetitive stimuli, which was most likely the case here. And they are opposed to the 22 kilohertz vocalizations, which are associated with negative states. However, those were not present in my study at all. So now starting the behavioral results part, we did not observe a correlation between individual reactivity to tickling and during drug-associated context response measured in the number of appetitive ultrasonic vocalizations. It probably means that the initial ability to express appetitive states in response to a natural reward isn't a good predictor of the response to a context conditioned by a pharmacological reward. However, the behavioral analysis confirmed the effectiveness of the conditioning model overall. So rats from the morphine group, here you can see them, emitted significantly more 50 kilohertz ultrasonic vocalizations compared to the cyan control group. Most importantly, some homogenetic manipulations in the amygdala were found to effectively modulate this response. So inhibition of serotonergic system, alone or with glutamatergic activation in the amygdala, significantly reduced the number of USVs emitted in the context response test. Whereas a simultaneous activation of both of these systems significantly increased it. So that shows that serotonergic and glutamatergic co-transmission in the amygdala plays an important role in the modulation of the affective component of the morphine-associated context response, and neuroadaptations occurring within this system, serotonergic and glutamatergic pathways, in the amygdala during the withdrawal period constitute an important element of mechanism underlying the phenomenon of incubation of drug craving. So here is the summary of effects of homogenetic manipulations on the ultrasonic vocalizations. Please have in mind that I might refer to the 3 groups from now on as serotonin on 5-HT- or glutamate or GLU+, purely as a shortcut. I will still mean the whole technically specific homogenetic process of activation and inhibition they indicate. So, due to the large number of variables, we chose to use Boruta algorithm to perform the neurochemical analysis, which allowed us to identify sets of features that are statistically significantly related to observed phenomenon. So in case when the phenomenon were the ultrasonic vocalizations number for all the groups that received morphine, Boruta identified the MHPG in retroscranial cortex, glutamate and DOPAC in the medial prefrontal cortex, and serotonin in the nucleus accumbens as relevant sets of neurochemical predictors. We did also the analysis for specific groups to see whether the neurochemical predictors will change. And for groups without the modifications of the serotonergic system, the identified key neurotransmitters were glutamate in the amygdala, striatum, and the medial prefrontal cortex. And interestingly, for groups without modifications of the glutamatergic system, we identified also glutamate, in this case in the striatum and retroscranial cortex, and also sarcosine in nucleus accumbens. So these differences confirm that the chemogenetic manipulations in the amygdala induce complex multistructural changes in the profiles of the 16 tested neurochemical analytes. But the focus of our interests were on the 3 groups with significantly different affect measured in ultrasonic vocalizations. And in this case, there came a particularly interesting and quite frankly puzzling discovery, because when we took a closer look at these groups, we noticed that the 2 chemogenetic manipulations leading to the same behavioral effect had fundamentally different or even opposite neurochemical profiles. And moreover, one of these groups appeared to be neurochemically consistent with the group that vocalized the most. And here in the table I'm showing you, you have only an illustrative fragment of the analyzed results, but this trend continued throughout the data. So, when I took even closer look at the neurochemical profiles of the 5-HT- group, they seem pretty logical and consistent with the observed behavior when I compared these results to the literature data. So the general pattern, decreases of serotonin, dopamine, norepinephrine across structures, and decrease in glutamate and increase in GABA in the amygdala were linked before with affect suppression. So it was logical to observe less ultrasonic vocalizations with that pattern of neurochemistry. In this situation, the question was what happened to the GLU+ 5-HT- group, because with increases of dopamine and possibly noradrenaline, it seems to have a neurochemical structure that should facilitate or make the animals prone to increase the affect and therefore increase their emission of ultrasonic vocalizations, yet the behavioral output remained exactly the opposite. And of course, when the human eye is not enough, you do brutal analysis. So this is indeed what we did. And the comparison revealed rather expected range of neurochemical changes in many structures that significantly differentiate these groups. So this first led us to the conclusion that the same behavioral effect may be achieved by different neurochemical pathways. And it did confirm the relevance of the GABAergic system, dopaminergic system, noradrenergic system, and glutamatergic system. So the reduction in affect in the 5-HT- group likely resulted from a combination of a global reduction in serotonergic transmission, a decrease in dopaminergic activity in the reward system, and decrease in norepinephrine and potentially increased GABAergic inhibition in the amygdala. And we got our attention shifted to glutamatergic system. So perhaps, despite the monoaminergic profile of the behavioral responses in the GLU+ 5-HT- groups, they were silent due to something overriding the monoamine systems. And maybe it is because of dysfunction caused by excess glutamate, which led to override. So naturally, we did also the comparison of the 2 groups with similar neurochemical pathways, but with opposite behaviors. And there were again signs of monoaminergic pro-affect increase to facilitate the increase of USVs. However, there were also present increases in the glutamatergic system, though not as high as in the other group, but still the question on what exactly happens in the other group, the GLU plus 5-HT minus group, remain not fully closed. And the answers were captured once again by the Boruta comparison. So among several features significantly differentiating these T groups were the levels of GABA and noradrenaline in the medial prefrontal cortex, the serotonin metabolite in the amygdala and other structures, and glutamate in the hippocampus. And still, please have in mind that additionally in the GLU plus 5-HT minus group, we observed the highest levels of glutamate in all of the studied structures, which combined with inhibition, maybe there was something there. So given all the data, it seemed that the increased emission of the ultrasonic vocalizations in the GLU plus 5-HT plus were the result of somewhat synergistic simulation of key components in the system, so an increase in dopaminergic activity in the reward system with a simultaneous decrease in GABAergic inhibition in the medial prefrontal cortex, which may enhance the positive affective state or reduce anxiety. Whereas despite the quite similar profile, in the GLU plus 5-HT minus group, the very high levels of glutamate observed in all structures could have led to dysfunction of the neural networks, perhaps through overexcitation or even excessive toxicity. I'm sorry, I knew this word was coming, which impaired the ability of key structures, such as, in example, medial prefrontal cortex, or maybe others, to properly express the affective state or process information. So all of this highlights the possibility that the similar behavioral effect can have different neurochemical substrates. Okay, ultimately, the correct answer to the question about the mechanism underlying the affective response to the context associated with morphine administration is somewhere in this graph, or even somewhere beyond it. This is a graph of neurochemical correlations of all the analytes we measured in this study in the 7 selected structures, illustrating the enormous complexity and interconnectedness of the studied neurochemical network. And it is worth to mention here that we were able to compile as many as 112 control points, so pairs of structure and analyte. But is it that many on a whole brain scale? I will leave that answer to you. It is important to remember from here that that manipulation of a, even one single point can induce a very broad effects. And some or more of changes in the network may ultimately determine the direction of the behavioral effect. And I would just like to point out that the real graph looks actually like this, because for the purposes of this presentation in the illustration shown on the previous slide, I have enlarged the notes to make them visible, but the exact and all of the correlation paths you can trace only on this one. And finally, the catfish. So, as I mentioned at the beginning of my presentation, we also performed the catfish method on the selected 7 structures of the brains. I really don't have that much time to explain the catfish in details, but it is a cellular imaging method which capitalizes on the different structure of 2 immediate early genes, ARC and Homer 1A. And because ARC mRNAs is derived from a short primary transcript, whereas the Homer mRNA is generated from a longer and more complex primary transcript, when double-labeled with specific ribopores, one can separate neurons activated at the start and at the end of the 20-minute context response test. And so these are only the very preliminary results, but we did get from them a few significant predictors for the behaviorally interesting groups. And we can observe that, for example, in the serotonergic inhibition group, the 5-HT minus, we had a reduction of affective state, and the activity of the infralimbic cortex, the retroscranial cortex, and prelimbic cortex was lower. However, a small subset of specific neurons in the disc granular retroscranial cortex were active throughout the context response test. So how this relates to the neurochemical profiles I mentioned earlier, we will find out as we will continue our work to obtain more catfish results and do even more analysis. So at this point, I want to conclude. Our findings strongly support the important role of serotonergic and glutamatergic co-transmission in the amygdala in modulating the drug-associated context response measured in appetitive ultrasonic vocalizations. We found out that the inhibition of serotonergic system in the amygdala alone or with combination with activation of the glutamatergic system in the amygdala results in the significantly decreased number of ultrasonic vocalizations, whereas simultaneous activation of both of these systems in the amygdala results in the increase of ultrasonic vocalization emissions. For each of the groups, we observed signature neurochemical patterns. And for the 5-HT minus group, affect suppression likely resulted from a combination of a global reduction in the serotonergic neurotransmission, a decrease in dopaminergic activity in the reward system, a decrease in norepinephrine, and potentially increased GABAergic inhibition in the amygdala. For the GLU plus 5-HT minus, the very high levels of glutamate observed in all structures could have led to a dysfunction of the neuronal networks, possibly impairing the ability to properly express affective state despite monoaminergic arousal. And for the GLU plus 5-HT plus group, increased affect likely resulted from a synergistic stimulation of key components of the system, such as an increase in dopaminergic activity in the reward system and the simultaneous activation in GABAergic inhibition in the medial prefrontal cortex. So, also, preliminary catfish results, catfish analysis results revealed that for the 5-HT minus group, the activity of the infralimbic cortex, infralimbics, retroscranial, and prelimbic cortices was lower toward the end of the context response test. However, a small subset of specific neurons in the disc granular retroscranial cortex were active throughout the duration of the context response test. So taken together, all these findings suggest that serotonergic and glutamatergic co-transmission in the amygdala might be a potential therapeutic candidate in search for treatment of addiction. Okay, I want to thank now everyone involved in the study, especially Adam Hammett, who is also a main organizer, as well we know, of this conference. The presented research was done thanks to funding obtained from the National Science Center. So thank you for your attention.